The goals of this ELISA-based method are first to determine the binding affinity of a cytokine to its receptor, and second, to measure the blocking effects of inhibitory peptides of the ligand-receptor interaction.
This method can help answer key questions in the field of protein-protein interactions, such as cytokine receptor binding. Binding affinities and blockade of these may increase the general understanding of interaction dynamics.
The main advantage of this technique is it is easy to perform, it generates reliable results, and it is relatively cheap due to the easy access of readily available equipments such as ELISA reader. Though this method can provide insight into cytokine receptor interaction, it can also be applied to other systems such as general protein-protein interactions and the designing of blocking compounds to inhibit the specific interactions.
Prepare the solutions as described in the text protocol. Make sure to filter the carbonate coating buffer with a 0.22 micron PES membrane. Use a vacuum. Also, make 10 different concentrations of each ligand or peptide. There are 2 assays described in this video. The first is the direct ligand-receptor interaction ELISA. It can be used to measure the receptor-ligand dissociation constant, which represents receptor-ligand binding affinity. The second assay is a recently optimized competition ligand-receptor interaction ELISA. This allows for the screening of peptides and other blocking compounds which act to interfere with the